is a fan of the Flames, but his second favorite team is the Jets. And he's actually got a jersey to prove it. Did his PhD in biomedical engineering at the University of Calgary, and then he and I actually overlapped during our time in Baltimore at Johns Hopkins University. I believe, if I'm not mistaken, he was the president of the Postdoc Association. And while I was there, all around good guy, and then stayed on as well in biomedical engineering there for a few additional years, doing some really interesting cardiac modeling and so on, which he's continued now into his faculty position at the University of Washington. So really looking forward to your talk today and hearing about what you've been up to, and go ahead and take it away.
All right, I'm gonna get the screen share going here.
Just, I will say, if everyone could, on the subject of bandwidth though, if everyone else could turn off their cameras for the time being during the talk, that would be great. And then at the end, during the question and answer period, then we can maybe all turn our cameras back on.
I'm trying to get the Zoom window to go to my other desktop. Can you see, are my slides showing up?
I can see it.
Okay, good. Perfect. All right, well, I can't see any of you, so don't say anything mean about me or make any faces. The title of my talk, as I submitted it to Chase, was using computational modeling and simulation to anticipate catastrophic future events. I realized that I probably should have clarified I meant future cardiovascular events and not earthquakes or typhoons or anything like that. As Chase mentioned, I am Canadian, and I tried to find a picture of me in Winnipeg, but I couldn't. I was there at a MyTax meeting once upon a time, and I loved it, but I found this one instead from the Heritage Classic in 2011, and that's my brother and I at McMahon Stadium. So I'll come back to the Made in Canada thing in a bit, but for now, greetings. Before I talk about the scientific component, I do want to make a quick plug. One of our assistant professors in the department is Kelly Stevens, and she published an incredible commentary together with collaborators at other bioengineering and biomedical engineering programs here in the US. This was published on Tuesday this week in Cell, and I really encourage you all to take a look. It talks about the disparity in funding between Black scientists in our field and others. And from the NIH. And I think, you know, obviously you aren't all thinking about the NIH on a day-to-day basis, but I still think this is a really important thing to have on our minds as bioengineers in terms of our commitment to what goes on in our field outside the scientific realm. So the overview for today's talk looks a little something like this. I'm going to start by giving you a primer about the computational modeling and simulation of cardiac physiology that my lab does. And then I'm going to tell you about some applications that we're excited about, both from my time at Hopkins and from my current work here at UW in Seattle, to translate this technology to the bedside. And then finally, I'm going to talk about sort of a research thread that I've been tugging on since 2011 or 2012 about using optogenetics as a means of defibrillating arrhythmias instead of electric shocks. So the virtual heart. Well, why would we model and simulate the heart first and foremost? This is a question we get frequently. Of course, powerful computing resources are now abundant. We have a nice, huge supercomputer here in Seattle, and we also have strong connections to the local companies that are doing cloud computing. There are detailed mathematical models of the relevant biophysical systems, some of which I'll talk about later in this lecture, and those are becoming more widespread, including for different species, different tissue and cell types, and different pathophysiological states. And of course, there's success stories from many other disciplines. So, you know, aerospace engineering is the first one that springs to my mind. You know, for decades now, before a single bolt is put in a piece of sheet metal, these airplane designs are subjected to hundreds, if not thousands or tens of thousands of hours of analysis using finite elements. And we wouldn't feel safe even putting these things in the sky if we didn't know that that had been followed. The same applies to most aspects of structural mechanical design. Moving into the bioengineering realm, we have success stories from computational genomics, computational neuroscience, systems biology, my personal favorite, computational epidemiology. And this is a paper that I learned about years ago at a MITEX meeting from Dr. Robert Smith. I'll let you read the nitty-gritty details as well, but I believe he's still at the University of Ottawa. In any case, we think that there's a lot of room to make this kind of discovery and to get these kind of insights from physiological models of the heart as well. So what systems are we talking about today? Well, this is a living animation from Wikipedia, one of my favorites. This shows the contraction of the human atria and ventricles. And the main thing that I want you to focus on here is the phenomenon as sketched in cartoon form here of cardiac excitation. So that precedes by a few, 100 milliseconds or so, the contraction of the atria and the ventricles. And it's sort of a critical component in terms of ensuring the delivery of blood to the body. The electrocardiogram in the bottom part of the cartoon has all the principal electrical events, the P wave for atrial contraction, the QRS complex here for the contraction of the ventricles, and then the T wave for the relaxation of the ventricles. If there's no coordinated electrical excitation of the organ, that means there's no oxygen delivered to the body, and that results in sudden cardiac death. So cardiac arrhythmia is broadly defined as any disruption of the heart's intrinsic ability to regulate its own rhythm. The bright red dot here in the top of the right atrium represents the sinoatrial node, which is the heart's natural pacemaker. And anything that happens that disrupts or overrides the sinoatrial node is an arrhythmia. Some arrhythmias, of course, are less immediately lethal than others. Not every arrhythmia is going to lead to sudden cardiac death. The biggest example, which we'll talk about a lot in today's presentation, is atrial fibrillation, where the excitation and contraction of the atrial becomes uncoordinated. And so the ventricles can continue beating on, if not a little bit irregularly. And this poses no acute threat in terms of well-being, but it's highly correlated or associated with stroke because of the clotting of blood in the atria that can ensue putatively. So what I want to talk about in terms of the detailed modeling and in terms of how we understand the way arrhythmias work is this broad concept of reentrant versus focal arrhythmia. So in reentrant arrhythmia, the aberrant activity is sustained by these vortices of excitation. And we'll talk a lot about how those can form and how we can represent that computationally. And those can override the sinoatrial node. In contrast, some other arrhythmias, PVCs, and some types of entrainment arrhythmias are focal or triggered arrhythmia. And what that means is that a secondary pacemaker, sort of an auxiliary aberrant or ectopic pacemaker, can form somewhere else in the heart for reasons that are related to pathophysiology. And that can, if that beats faster than the sinoatrial node, then it can cause problems in the heart. From a computational standpoint, reentry arrhythmias are a lot more straightforward to simulate. And so that's what we do for the most part in my lab. And that's mainly because the events associated with driving triggered activation happen at the protein scale and include sort of heterogeneity at the cell scale, which is something that's difficult for us to represent computationally. So we're going to be dwelling primarily in the space of reentrant arrhythmias today. Now, there's still major challenges associated with modeling arrhythmia, even reentry. And what I'm showing you here is just meant to illustrate the fact that events in the cardiac excitation sequence are at centimeter scale in the spatial sense and second scale in the temporal sense. But if we look at the cell scale, things are happening. A single cardiomyocyte is tens or hundreds of microns long, and the time scale of events in these cells is on the order of a few microseconds, especially really rapid events like the upstroke of the action potential, which is shown here, or not shown things like release of calcium from the sarcoplasmic reticulum to precipitate engagement of the contractile filaments. And so that disparity in scale is what makes this a really challenging computational and mathematical problem in terms of having suitable numerical methods that can handle that divergence in scale. So I'm going to tell you a little bit about how we do that. At the cell scale, we can think about the individual myocyte as a chunk of membrane with these aggregated populations of different ion channels, sodium channels, different types of potassium channels, calcium channels, and subspaces. And we can lump that all together and represent it by solving a system of ordinary differential equation that represents the kinetics of the membrane. And this gives us realistic models that can simulate with a fair degree of accuracy cardiac action potentials. In this case, it's a human ventricular action potential model that I'm showing you. Those cell scale components can then be plugged into tissue scale building blocks. And specifically here, what I'm showing you is a schematic of what's broadly described as the bidomain formulation. And essentially what I need you to understand in order to proceed with this talk is that we can solve one or more PDE systems at the tissue scale in order to realistically represent the propagation of excitation in cardiac tissue. And this is just a really simple example showing a planar wave initiated by stimulating the bottom part of the slab here. For larger domains, this essentially has to be solved numerically using high performance computing systems, because otherwise it would be just too unruly and it would take too long on a desktop or laptop. So then finally, at the organ scale, we take the cell scale plugged into the tissue scale, and then we span a domain or deform a domain with representations of the intracellular and sometimes the extracellular space on a realistic cardiac geometry with or without representation of disease-related remodeling. And this is something that I want to take a moment to emphasize. So the top row here shows a canine model of myocardial infarction, and you can see that the people who did this research, this wasn't by me, but it was from one of our reviews in 2014. Basically, these green and yellow regions are areas of the heart that have been affected by the ligation of one of the coronary arteries to create an experimental model of a heart attack in these dogs. You can see the dense yellow scars surrounded by green peri-infarct or border zone. And really the key element of the way our models work is that we can change the properties of the tissue scale model and or the cell scale model in those regions delineated by enhancement that's extracted from images. And these are clinical grade images. These aren't ex vivo images in a 7T MRI scan or anything. This is canine imaging with a clinical grade scanner. And on the bottom row here, this is a human scan of the patient's atria with the red regions showing locations of fibrosis, which correspond to enhanced regions in the MRI scan. Okay, so the second challenge, once we tackle the issue of scale, is the nonlinearity of cardiac electrophysiology. And so I've got a short anecdote here to explain what I mean by that. So these are simulation data from a single cell action potential model. And if we stimulate it twice, 1 second apart, we expect to see two action potentials, which are more or less identical. But if we shorten the coupling interval between those two stimuli, things are going to go wrong. And specifically, because of the refractory period of the cardiomyocyte, we're going to see an abbreviated action potential duration. It's weaker and wimpier. It's got a slower upstroke, which is hard to see in this video, but I can assure you it's the case. And if we take that to its extreme, this can lead to complete suppression of the second action potential. So the absolute refractory period here is on the order of 300 milliseconds. And you see that second stimulus, it affects the cell. There's an artifact here, but it really doesn't produce a second proportional response. And that has major consequences at the tissue scale. So what am I talking about here? Back to our example of the planar wave. We initiate the wave. At some point, it's still refractory up in this, the north half of the slice, but in the south half, the tissue is recovered and ready to go again. It's already fully repolarized if you think back to the standard action potential wavefront. And if we deliver at that point a critically timed and located second stimulus, what that's going to do is initiate reentry. And so this is a classical pattern called a rotor or a spiral wave. In 3D, you might hear it referred to as a scroll wave. But this is what we're really looking to prevent in terms of the hearts of human beings, because it's self-perpetuating excitation, and that's a bad thing because it can override the sinoatrial node. And so being able to model that type of complexity is absolutely critical in the context of our work, because there are these two ingredients for reentrant arrhythmia, which are substrate. And in the previous sort of cartoon example, that's the state of the tissue after the first stimulus, the plane wave. So you have spatially heterogeneous distribution of regions that are refractory or ready to be stimulated again. And then the second ingredient is a trigger. And that's, in our example, the critically timed and located second stimulus. And what I want to impress upon you is that many pathophysiological conditions can give rise to exactly those types of proarrhythmic substrates and triggers. And the fact that reentrant arrhythmias are so complex and so difficult to wrap our heads around, even in a clinical standpoint or even in an experimental condition, means that simulations can really help us explain how they work and potentially reveal new treatments. So the conceptual basis that I'm talking about here is that if you take a healthy human beating heart or beating human heart, add substrate, which can come from disease, genetic predisposition, fibrosis is the prime example in our work, and then also add triggers, and that can be, most human beings can tolerate an ectopic beat that happens now and again. These are things that happen without, consequences. But if you have an ectopic beat, or if you have stress that causes additional burden of ectopic beats, and you combine that with substrate, it can have lethal consequences. And this is just an animation showing an example of a heart viewed epicardially and endocardially in ventricular fibrillation. So how can we use these physiological models? Well, my lab has tried to explore applications ranging from basic science to translational. And to get a little bit more specific about what I mean, we can get mechanistic insights, given certain circumstances or genetic mutations, how does an arrhythmia happen and why does an arrhythmia happen? We can develop novel therapeutics, such as new drugs or new devices. We can take an MRI scan, run simulations, and then try and approximate an individual's personalized risk based on virtual arrhythmias observed in silico. Or at sort of the apex of our sort of translational pyramid, we can take an MRI scan prior to a clinical procedure designed to ablate using RF catheter energy the specific tissue region responsible for arrhythmia. We can take an MRI scan prior to that, run simulations, and then use the information from the simulations to actually steer the clinician's decision-making during the procedure. And I'm going to tell you about the example that we've done in a pilot study for that as a case study in the rest of this lecture, or the second half of this lecture. And there are different parts of the lecture that correlate to each of these. And so we'll come back to this at the very end, and I'll tell you how the examples I've given you map onto these different applications. So modeling the atria. And, uh-oh. I still have everybody?
Yeah, we're still here.
You're still here, but the slides have died. I certainly do. Most of you probably know someone in your extended family who has atrial fibrillation. This is an enormous public health problem worldwide. It's the most prevalent sustained arrhythmia. And as the median age of the population on Earth increases, as we live longer and relatively healthier lives, we're all going to get old hearts, and old hearts have a tendency to fibrillate, especially in the atria. And so accordingly, the best evidence that we have is that the prevalence will increase 2.5-fold by 2050. It's a huge, huge number of people who have this arrhythmia. And as I said earlier in the presentation, it's not going to kill you immediately. It's going to give you this irregular ventricular heart rhythm, but it's associated with a dramatically increased risk of stroke, which can be catastrophic. And what we really don't want to see is transition from paroxysmal AFib, where you get it now and then, to persistent AFib, where episodes can last a week long or more uninterrupted. And the reason why that's so critical is that catheter ablation, which is a treatment where a catheter is inserted through the groin, navigated up to the heart, and used to essentially cauterize tissue that's thought to be responsible for perpetuating AFib, the long-term success rates for those procedures are extraordinarily low once patients have transitioned to persistent AFib. And we know that there's a relationship between fibrotic remodeling, so collagen deposition, fibroblast proliferation, and myofibroblast signaling. We know that there's a relationship between that and these reentrant drivers of atrial fibrillation, but it's not really well understood. And so when we embarked upon this research back in 2015 at Hopkins, our central thesis was, computational models can be a huge help in understanding this. So this is a story of reconstructing models from late gadolinium enhancement MRI. And so the gadolinium binds more tightly to the collagenous tissue, to the fibrotic tissue. And so when we segment the scans, we can identify the hyperenhanced regions and tag those as fibrotic to be treated in the model. We then map an atlas version of the human atrial fibers diffeomorphically into the patient-specific atrial geometry. And this is because imaging technology hasn't come along far enough yet that we can properly resolve fiber orientations. In order to represent the electrophysiology of these different regions, we incorporate human atrial models of non-fibrotic and fibrotic myocardium. The key differences here are that fibrotic tissue essentially has a slightly weaker upstroke. It has a slightly depolarized resting membrane potential because of the ionic consequences of fibrotic remodeling. And then the really important thing is that it has a longer tail, meaning that the refractory period for fibrotic myocardium is a little bit longer, and that can lead to conduction block, and in the long term, create a substrate for reentry initiation. And that's exactly what happens when we impose a virtual cascade of electrical stimuli from many different locations in the left and right atria. We end up seeing the initiation of rotors, and this pattern of vortex-like excitation that you're seeing here in the spatiotemporal evolution of membrane voltage in the models should be reminiscent of what I showed you in the introductory slides, this rotor pattern. And we're looking here at the superior vena cava of one of our patient-specific models. Okay, and when we collapse that down to look at the activation times, oops, we're going out of control here. We can see that the path of the phase singularities, which my advisor who at Hopkins, who lived through Katrina in New Orleans, always called these the eyes of the hurricane. So the eye of the hurricane sort of meanders back and forth, and these are relatively compact regions that can be targeted with catheter ablation. This is on the order of 0.5 centimeter, maybe, on the scale of the atria. And the really consequential observation of this early mechanistic work was that, for each of the patients in our cohort of all persistent AFib patients, we only found, maybe 3, maybe at most 6 of these pro-reentrant driver harboring regions. And this is important because if a clinician wants to go in and try and make a fibrosis-based decision on where to destroy the tissue in order to prevent more AFib, what our work suggests is don't worry about this fibrosis over here. It's less of an issue than these 4 key regions. Those are the ones that really need to be focused on. And so armed with that mechanistic knowledge, we embarked upon a long-term translational effort to do a pilot study of 10 individuals at Hopkins recruited sequentially to the study, where we would do exactly what I talked about earlier, get them an MRI scan a couple weeks before their ablation procedure, run simulations, and use those to decide where the clinicians ablate. And I only have time to show you a couple examples. This is one of our latest patients. This was 9 out of 10. I should add a note there. This is the reconstruction from their MRI scan. Interestingly, in this case, we could only see a different type of arrhythmia called atrial flutter. And so these are activation maps, just like I showed you in the previous slide. And what you'll observe is this anatomical region here. This is the mitral valve. So underneath this, in the full heart, would be the left ventricle. And you can see that the activity is sort of doing ring around the rosy, either counterclockwise or clockwise. And this is a very well characterized arrhythmia called left atrial flutter, atypical LA flutter. But it poses a problem for us in the context of simulations, because this isn't the type of thing that we're trying to ablate for terminating persistent AFib. And so our solution was to do virtual ablations in the model. And I don't want to belabor this point too much, but essentially what we would do is run simulations, ablate the models in silico without anything ever happening in the patient, and then try and re-induce the arrhythmias, rinse, lather, repeat until we had a completely non-inducible model, and then export the observations into the mapping system, which is called CARTO. It's a Biosense Webster product, and that's what they use to guide these procedures in real time, these cath ablation procedures. And so sure enough, when we ablated, and you can see the big thick golden lines of simulated RF ablation in our models, when we ablated this particular model, we saw the emergence of reentrant drivers in the posterior left atrium here. You can see the sort of clearly defined vortex pattern. Hopefully the video is coming through all right. And then here's a second reentry morphology that's in a similar region, but closer to the right inferior pulmonary vein. And so on the day of the procedure, we loaded a personalized representation of this model, annotated with the information from our simulations into the clinical mapping system. And the clinician was able to, put this check mark shaped ablation lesion that was specifically designed to render that, that type of reentry impossible in the patient's heart. And this is not a usual line that they would do clinically. So this is very much guided specifically by the simulations. And what we saw was extremely exciting for this individual. So prior to ablation, they were in stable atrial fibrillation, which you can see here. These are the intracardiac electrograms from inside the coronary sinus of the patient. During the ablation of that sort of checkmark shaped target, the AFib terminated abruptly. This had been going for 20 minutes and it terminated spontaneously during the ablation. Okay, that's not totally surprising. That's the kind of thing that happens sometimes during RF ablation. But what we were really excited about was when the clinician tried to re-induce atrial fibrillation by pacing really aggressively. All of these plus signs and S1 labels, that's the clinician delivering an electrical stimulus in rapid sequence to try and get the atria to fibrillate. And in the end, AFib was completely non-inducible at the end of that procedure. So this isn't the be-all end-all. This is it's a step in the right direction, is what we said at the time. But overall, when we looked at between half a year and a couple years of follow-up in these 10 individuals who were recruited to our pilot study, we were excited to see that there was no recurrence of persistent atrial fibrillation, and only one individual enrolled in our trial had an AFib or atrial flutter ablation before the end of follow-up. So this study is ongoing, the next step here. I'm still involved with this, but recruitment is happening right now at Hopkins, and I put the NCT here so that you can check it out on clinicaltrials.gov. So the takeaway messages for this case study are, we have a proof of concept driven by this mechanistic knowledge from our prior studies for a new treatment paradigm in catheter ablation of persistent AFib. We did have to overcome, I didn't get a lot of time to talk about this, huge technical and logistical challenges, especially in that step where we were loading the personalized ablation plan into the clinical mapping system. We did that all by ourselves. We didn't have any help from Biosense Webster. And frankly, I don't think they were terribly pleased with the fact that we were trying to use their hardware for this, but we got it to work anyway. we have cautious optimism about the clinical outcomes. And the trial that's ongoing, it's a randomized trial. It's cleared by the FDA. There's an investigational device exemption for it. And so hopefully, fingers crossed, we'll have good news for you within the next few years regarding that. And that will be the ultimate test of efficacy for this type of procedure. So the second part of the talk is about atrial fibrosis in patients with ESUS, which I wouldn't be surprised if none of you have ever heard of it. I certainly hadn't before I started working on this. That's embolic stroke of undetermined source. So I left a typo in here. It should be undetermined, not unknown. And essentially that's stroke in the absence, it's stroke that looks like the type of stroke that is caused or that is associated with AFib, but the patient has no evidence of AFib. So they get Holter monitors, they get Zio patches, they get all sorts of ambulatory monitoring, and there's no evidence of atrial arrhythmia. But the fascinating thing about these patients is that, and this is from a 2019 study by my collaborator Nazem Akoum here at UW, they have the same extent of that fibrotic remodeling that is so intrinsically connected to both symptoms in patients with AFib and also outcomes of catheter ablation procedures. People with more fibrosis tend to do worse in ablation procedures. And so this is a real paradox because those ESUS patients, this middle box and whisker plot here, they don't have any arrhythmia, and yet they have these very fibrotic atria. Okay, so how can we help unravel this mystery with modeling and simulation? Well, we think we can try in the long term to determine which ESUS patients will have a recurrent stroke, but we don't have the good modeling tools for that yet because we think we need to model hemodynamics and biomechanics in order to do that. So that's TBD. But what we think we can do is try and figure out which ESUS patients might be at risk of future atrial fibrillation. And to put a fine point on that, we also have this interesting mechanistic question, which is if they have so much fibrosis, why don't they have atrial fibrillation? And this is something where we think we're really well set up because we can test two possibilities. The first is, okay, maybe there's something fundamentally different about the pattern of fibrosis. We know from our previous work that pattern can play a huge role in addition to the extent of fibrosis. Pattern can have a huge role to play in terms of where rotors localize and whether rotors happen. But the second possibility, which I'll spoil this, we think this is the case, is that fibrotic substrate is the same between these two individuals, but there's a lack of arrhythmia triggers in the ESUS population. And so we think simulations are a perfect tool for helping to clarify this. So we recruited an extremely large cohort, 45 patients between here in Seattle and our collaborators in Germany. And we matched those against a set of 45 AFib patients at UW in order to facilitate comparison. We developed a completely new atrial modeling framework for this, which is fully automated and involves reconstruction of only the left atrium, which makes it feasible for us to go from pre-segmented images to fully analyze results of our in silico stress test in about 4 hours. Oh, this is interesting. Okay. So I think I see what happened here. When the computer crashed, I loaded up the wrong version of this slideshow. Hang on a second. I can take questions while I'm fixing this. 1 second, I want to fix this. Chase, are you still there?
Oh yeah, we're here.
Okay. Sorry about the disruption.
No worries. I think with all the virtual stuff, everybody's probably used to this by now, so no biggie.
Okay. Oops. This is I usually do really well in virtual talks. This is very embarrassing. Okay, better. All right, that's why the typo is still there. That was the bellwether typo, I should have known. Okay, so here are 45 ESUS models. Here are 45 AFib models. To the naked eye, if I were just looking at this, I would say, nope, they look about the same in terms of pattern, in terms of burden, which ends up being true, but we didn't know that at first blush. And so we ran a bunch of simulations. And what we saw was, in the AFib models, unsurprisingly, these patients all had arrhythmia. we saw that reentry was initiated in many of the AFib models. That's not a surprise at all based on our previous work. But what is somewhat surprising is that we also saw inducibility in the virtual framework of reentry in many of the models for patients with ESUS. And so what's going on here? Okay, so we observed, this is the sort of big picture summary slide, about half and half from the two populations were inducible, and the proportions didn't look to be that different. It's not a huge surprise that there's some non-inducible AFib models, because like I told you early on, fibrosis-mediated rotors are just one of many potential mechanisms for arrhythmia perpetuation in these individuals. So this is about what we expected to see for AFib. But for these ESUS models, what we're really curious about is, okay, well, is there some obvious difference in the fibrotic remodeling? So we know that there's no difference in burden in the inducible population, but what about the other attributes? And so, the next set of slides are all about the things that we ruled out. So, inducibility, this is just the same data presented in graph format, no difference statistically. This is a plot showing for all 90 patients in the cohort, both the inducibility status, circle being yes, it was inducible, triangle being no, and blue versus red, ESUS versus AFib. The only interesting thing here is that we had some ESUS models with remarkably low fibrosis scores, less than 10%, that were still inducible, which is somewhat remarkable. We haven't really dug into that yet, but it's sort of on our list of things to do. In terms of the atrial fibrosis burden, sort of consistent with our prior study, no statistically significant difference in the subset of models that were inducible for the AFib versus ESUS cases. And this was sort of our major top line conclusion here, no major differences in the extent of fibrosis or in rates of inducibility. Likewise, when we looked at the number of pacing sites, virtual pacing sites that induced arrhythmia, no difference. In terms of the number of unique locations in each model that could potentially harbor these reentrant drivers, no difference. And so our fine-grained inducibility analysis had the same conclusion. AFib and ESUS are indistinguishable in terms of the intrinsic potential of the fibrotic substrate to sustain reentry per se. So that's essentially what I just said there. That's our first key takeaway from the sub-study. The most important result of our work here is not a finding, but in fact a new hypothesis. And that hypothesis is the lack of arrhythmia in these patients with fibrosis could be due to a lack of suitable arrhythmic triggers. And so we have sort of started to percolate ideas on how we could test this, put Holter monitors on these patients and look for premature atrial contractions and see if there's a differential between the ESUS and AFib populations. And we expect there will be, but there's more to come. And the last I'll say is, you can find our preprint on this subject, which summarizes a lot of the data that I've included here on medRxiv. And I guess the true last note is that, as I mentioned earlier on, we'd really like to know more about, we have all these images, we'd really like to do patient-specific risk stratification for recurrent strokes, because one of the challenges with ESUS is that we don't know if there's a good medical argument for medicating them, for giving them these novel oral anticoagulants, which are super expensive and can have major side effects. And so there's a real case to be made for computational risk stratification. And in order to do that, Nazim and I are partnering with a new professor here, Juan Carlos del Alamo, in mechanical engineering. And we're going to try and develop these multi-physics models. And you can read about our vision for that in this recent paper published in Heart, that you can find. Okay, so part 3. And this is about optogenetic defibrillation. So if we were in a classroom, I would ask how many of you have heard of defibrillation, but since we're on Zoom, I'm just going to give you a quick synopsis, Optogenetics 101. So this is the genetic encoding of light-sensitive proteins. So these are exogenous proteins, they're from algae, and we can encode them into excitable cells. The most common one is channelrhodopsin 2, and this is, de rigueur these days in neuroscience studies. Maybe it's even old hat. But the idea is that when we express channelrhodopsin or other opsins in excitable cells, we can then apply light stimuli, irradiation, in order to elicit some kind of effect. And so in the case of channelrhodopsin, it elicits a large depolarizing current. It's not quite as large as the intrinsic fast sodium current, but it's good enough to give you a nice action potential. And so these are simulation data from my 2013 paper, which was sort of a response. It was our idea of like, wow, this field is just, it's just coming to life. The first experiments were done in 2010 in cardiomyocytes expressing channelrhodopsin, and that followed about 5 years after the real explosion of neuroscience optogenetic research. So these are sort of micrograph views of channelrhodopsin expressing cardiac tissue in mice. And then on the right-hand side, that's a Langendorf perfused mouse heart being entrained to a faster beating rate using light focus through a macroscope. And these are from my collaborators lab, Philip Sasa in Bonn. And so our response to this was, well, we need to plug this into computational models. This is fantastic. And we knew that there were biophysically realistic channelrhodopsin photocurrent models out there. And we knew that it would not be that challenging for us to incorporate things like realistic patterning of light sensitive cells and an attenuation model to represent the loss of light intensity across the myocardial wall if we're illuminating at the epicardium or the endocardium, things of that nature. And so we published this and started to do new research. Could this work? Could we defibrillate a human heart? If we can get if we can get it to express channelrhodopsin, can we defibrillate it with light? And so this gave rise to, I think this is one of our greatest collaborations to date on this. It's side-by-side experiments and simulations. So this was again with Philip's lab at the University of Bonn, and they did perfused mouse hearts. They attempted defibrillation of ventricular tachycardia, and we tried to match that in a simulated human heart. And so lo and behold, when they applied the light stimuli to the hearts that had been put into tachycardia, they saw a high rate of optogenetic defibrillation success. And when we went into the simulated human hearts, so this is a video showing the basal tachycardia in this patient-specific model. Here's the same simulation repeated with blue light stimulation. Okay, it didn't work. And that was a surprise to us somewhat, but we knew if you look here at the silhouette, this is the sort of the directly stimulated region, you can see maybe 20% of the thickness of the right ventricular wall, and that's the thinner of the two ventricles, was affected. And so this was not super surprising to us, but we were, of course, disappointed. But what we thought was, okay, this is an attenuation problem. There's photon scattering and energy absorption across the wall, and we know that red light can penetrate a lot deeper. And the motivation for this was, at the time, there was a ton of research happening in the neuroscience optogenetic world to develop red light sensitive alternatives to channelrhodopsin. And so when we attempted that, you can see the direct effects of the light are much deeper in the right ventricular wall. And sure enough, we were able to successfully defibrillate these arrhythmias. So in the very last slides here, I'm going to talk about a new project, which is the use of an inhibitory opsin, which is Giardia theta anion channelrhodopsin. And this is meant to induce repolarizing current in response to green light instead of depolarizing current in response to blue light. And this is data from the original experimental studies by Govorunova. We were able to incorporate a simple two-state model that realistically recapitulates the behavior of that light-sensitive ion channel when subjected to green light illuminations. And when we plug it into atrial or ventricular myocyte models, we see that initially, when we apply a light pulse here at the beginning of the illumination onset period, that causes a transient depolarization towards the resting potential of these channels, which is around -40 millivolts, so these traffic chloride ions. But then once the initial transient settles down, we have a pretty solid voltage clamp to that potential. And despite attempts to stimulate electrically, which are shown here by the second star, these cells aren't going anywhere other than staying at that -40 millivolt resting potential. Okay. So looking at the results in the organ scale simulations, so these are attempts to defibrillate atrial arrhythmia using either blue or green light. Unsurprisingly, given the nature of these arrhythmias and the relatively weak illuminations used in this simulation context, they were unsuccessful. You can see that there's some subtle effect here in the video. I think it's probably hard to appreciate over Zoom, unfortunately, but it's not really the point anyway. The point is that when we had this much beefier and stronger green light sensitive current, even though green light penetrates more poorly in the myocardial wall, because the current in these GTACR1 channels is so much more powerful than the current in the channel-rhodopsin models, this was good enough at a very low irradiance of 0.1 milliwatts per millimeter squared applied to the endocardium to terminate the arrhythmia. And, you know, unsurprisingly, the mechanism is this direct voltage clamping. And you can see even across the left atrial free wall, which is on the order of a few millimeters wide, we still see a pretty reliable abolition of nearly all of the electrical excitation in the context of endocardial illumination. And this was feasible. So what I showed you was 0.1 milliwatts per millimeter square. Even several orders of magnitude lower than that, we were still able to see successful defibrillation, which it raises some really exciting possibilities for future potential translational opportunities. And here's a tabulation of all of our experiments in 3 different atrial models, different irradiance strengths for blue, green, and red stimulation. So our conclusion here is that optogenetic defibrillation of these atrial arrhythmias, this is the most feasible thing we've seen in the space of optogenetic defibrillation, in my opinion. If we can find a way to get the heart to express these opsins, if we can find a way to light up the inner surface of the heart using some kind of, you know, fiber optic device or biocompatible flexible material, then I think this would work. And the reason why is the energy requirements are, you know, orders of magnitude lower than with the sort of, you know, nearest competitors. Our preliminary data suggests this could also be effective for defibrillation in the ventricles. Of course, this would require getting a light source inside the left ventricle, which would probably lead to all sorts of problems. And so I'm not ready to really talk about that yet. Maybe it would be better to try and target the epicardium, where we could do some sort of pericardial illumination or something like that. But that's for a future time. So optogenetic defibrillation, we think it could work. We're optimistic. And I mean, this is super future gazing stuff, and it's certainly not going to happen next week, but it could work, at least theoretically. But we know that light attenuation is a huge constraint. We think that computational models can guide new experiments. And what I really want to stress is that, you know, a lot of the work that we did early on with Philip they were on the precipice of trying the same type of defibrillation experiments in rabbits or pigs as they were doing in mice. But our results from these computational studies gave them pause and said, like, well, if we really are looking to translate this, we need to think about ways to tackle this, you know, transillumination attenuation problem before we move to larger preclinical animal models. And so to my mind, there's a sort of, you know, there's an experimental resources and like an animal rights issue that is addressed by this. Is the simulations are all doable, at least theoretically, without ever lifting a finger in the lab. And so we think, of course, there are huge hurdles to translating this technology, but our new computational tools, we hope, will be very helpful to the community in terms of figuring these things out. Okay, so take home messages. Back to the slide I promised to come back to. So we talked about, you know, the spectrum of basic science to translational applications. And I hope that I've persuaded you through today's presentation that the lab is tackling the whole spectrum of these areas using the physiological modeling tools, ranging from learning more about drivers of persistent AFib, discovering the potential for optogenetic defibrillation, looking at the risk of potential arrhythmia in patients who have a stroke but no arrhythmia but might have arrhythmia in the future, and finally, the personalized ablation procedure planning for individuals with persistent AFib. So that's the work. This is the beautiful cartoon of my lab. The people who did the work are a couple of the PhD students, as well as one of our brilliant undergraduate researchers, Griffin. And I think that's it. And it's only 2:51, so not bad.
Excellent. Well, thank you very much for a very comprehensive talk. So I think what we'll do for questions probably is I think everyone can go ahead and turn their cameras back on so we get a little bit more personable while you're asking your questions, if you do have a webcam. And I think in order to take the questions in order, what we had done last term was for people to just raise your hand or type question in the chat, and we'll sort of go in order that way. So questions in the chat. While people are doing that, maybe I'll just ask one off the bat. I was really curious. It was something that you mentioned just in passing about the diffeomorphic mapping in order to get your subject scans into sort of like template space. And this is something my lab's been really interested in with brain scans. And so I was curious what, if you could just discuss even very briefly about what approaches you guys have used and if you've like experimented with different approaches for that, because that's non-trivial, I presume.
Excellent questions. Yeah, so you caught me. Basically, the diffeomorphic mapping stuff that we used in the original study was LDDMM, which is
Okay.
large something
Yeah,
diffeomorphic.
Michael Miller.
Yeah,
Yeah.
so this
Yeah.
is Mike Miller, who was a professor at Hopkins in our department and
Yeah.
who's now the chair there. And so, yeah, for years we used LDDMM, and it
Yeah.
was total overkill. When we were doing the clinical trial or the pilot study, it was the Achilles heel of our computational workflow because we
Okay.
would get the images, we would build the FEA model, and then we would go to map the fibers, and we would have to wait like 24 hours. And
Yeah.
it was total overkill. And they kept telling us, like, you guys, what you're doing is way overkill for the problem you're solving. So Stephen Nieder's lab at King's College of London has developed an alternative workflow based on imposing a atrial coordinate system on the atria, and then using the basically, if we can take a patient specific atrium and then unwrap it into a Cartesian like grid and have a theta coordinate and a phi coordinate, then if we have the fiber mappings for the epi and the endocardium. So that's what we're doing for our new simulations, the ESIS simulations that I showed you. And that's part of why we can do it in 4 hours instead of waiting for the CIS supercomputer to kick in and do LDDM.
Okay, that is what I was wondering, because you did show later in the talk that you said it was like 3 or 4 hours total, and I was thinking, what are you doing for the spatial normalization then? But okay, thank
absolutely.
you. Yeah, thank you. Okay, so I see that now there are a few questions in the chat. So Susan, go ahead. I think you might still be muted.
I think you're still muted.
Oh, there. Now I'm unmuted. Can you hear me?
Yes.
Yes, loud and clear.
Oh, good. Often that doesn't happen. My camera and my voice conflict. So for the multi-physics modeling, I was wondering what software you used and what your reference was. You gave a reference for, you'd written a paper about the multi-physics modeling?
Yes, so that's a white paper that we published last month in, or well, maybe in, I don't know, maybe it's not even out yet, but it's in Heart. I will find a reference to the paper while we're talking and put it in the chat. So we haven't incorporated that. We haven't done that yet. That review was a vision for what we can do in the future. The EP modeling software that we use is freely available for non-commercial applications. It's called OpenCarp, and I will put a link to that in the chat as well. So let me.
Do you know anything about open foam?
I do know about foam. Yeah, I haven't played around with it myself. The issue with the carp stuff and the fact, so to give the backstory on carp, you know, I've been using it since it was a very closed source program in my advisor's lab at University of Calgary, and he had spent at that point the prior 10 years developing it. And it took until last year for them to get to a place where they could be persuaded by people like me to open it up. And now it's called Open Carp, but it's very much not open source. And that's, I think there's a tension there, and it's something that we could discuss. I could go on for hours and hours about this. It is free to use, but they're not ready at this point yet to open it up to the community for everybody. In any case, I don't know about the reason why we need specific tools for this is because the scale mismatches that we're dealing with in cardiac EP, I think that the solvers that we need to implement and the numerical sort of tricks that we need to pull are so specific to the cell scale cardiac EP models that it becomes very difficult to use generalized tools for that, generalized FEA at least.
Well, I'm interested in cellular level, so that would be fine with me.
Yeah, so CARP can definitely work at the cell scale, and all of the results that I showed you in the presentation today came from that. And like I said, it's the kind of thing where you have to send them an email and say, you know, I'd like to check out your software, and then they'll send you a binary to install it. Yeah, the documentation is a little shaky though. I have to say that's the thing. It's available, but it's not user friendly at all, which I think is part of their argument for why they didn't open source it a long time ago or even make it available, because the learning curve is catastrophically steep. Yeah, and there's a link in chat to the HART white paper.
Great, thanks.
Thank you.
You're welcome.
Okay, were there any other questions? Yeah, you can raise your hands as well, Zara had pointed out. So I guess in terms, I'll ask one more. I was really interested in that personalized, like designing personalized approaches for the ablation. And so obviously that's ongoing right now. It sounded like that takes like 3 or 4 hours. 3 or 4 hours on what, out of curiosity? Like, is this something that could be done on like Compute Canada or Amazon Web Service, or will this actually run on a standalone GPU workstation or something?
Good question. So for the risk strat stuff with the new models that we're building here from the left atrial, for the left atrium only, we're running those on supercomputer nodes that have, you know, between 28 and 32 cores each. I think maybe some of the newer nodes have 40. Okay. I have not fooled around too much with the cloud supercomputing stuff, just because in all of the time that I've tried several times, I've tried to get it going on AWS. I tried to get it going on Azure. I even had a fight with lawyers at Hopkins because I wanted to try to get it going on Google Cloud Platform, and they told me I wasn't allowed. Okay. Compute Canada, Westgrid would be the exact resource that if I were at a Canadian university, I would be all over that. And we did use Westgrid a lot when I was in Calgary.
Okay, but they will run, you were saying, on nodes with like 30 to 40.
Oh yeah. So
okay.
there's some real characters over in Europe who are trying to scale these simulations up to, like, you know, thousands of cores that are networked together with a sort of low latency backend fiber stuff, which is cool, you know. And the idea is like, okay, could we run a simulation of a heartbeat in the time it takes to actually have a heartbeat in a body, right? Could you run a simulated second in less than a second, which is theoretically possible, but it's just like, to me, that's not really that entertaining compared to the workload of getting it to go, and the idea of like marshaling those computational resources.
Right.
So for the clinical trial that's ongoing, the turnaround time is still quite a bit longer. So because they have to do the virtual ablations, that's manually intensive. There's some analysis steps that are intense. They also have to reconstruct the model themselves, whereas we're going from a pre-segmented MRI scan, which is part of the clinical standard of care for patients here at UW. But that's kind of unique, and it's like the feather in our cap. So that process, they ask for a week between the MRI scan and when the patient is going to go onto the table to get their ablation. And they were like, the regulatory hurdles were difficult. Like the FDA was very demanding, and I was part of that process. And it was really, I was impressed. You know, this is the kind of thing you want to hear, right? That you want to know that they're going to hire external consultants to scrutinize your computational work and make sure you're not, you know, trying to pull a fast one on anybody. And so we got through that, and then the pandemic hit, and I think at that point they were just about ready to start recruiting patients for it, but they had to shut everything down at Hopkins. And I still don't think they've ramped back up to full capacity for these procedures.
Yeah. Sure. Okay. Well, thanks very much. We're beyond 5 o'clock, so I think time to let people go. But that was really interesting.